First question, important. Everyone turn on camera. Everyone turn on camera. We are doing only the revision and which is important. This chapter is again exactly similar number of questions as life process has. Although it is conceptually it is easy, but the types of question asked on this are many more. So first thing, importance of DNA copying. Or first thing before that, what is reproduction and what is the significance of reproduction? What do you mean by reproduction? First thing. Reproduction notes I have already given you on what's this one. Google Classroom I have uploaded. Revision notes. So what is reproduction?
The process in which DNA from two parents combine to form a new individual organism is called reproduction.
Reproduction is a process.
Ma'am, it is a biological process where they give birth to their offspring.
So new organisms are produced. New individual of their own kind, own species are produced. So new individual of their own species. This word should come. The reproduction is a biological process in which every organism produces their new individual or new offspring of their own kind or own species is reproduction. Significance of reproduction?
Ma'am, it ensures the continuity of the survival of the species.
It ensures the continuity of species. So one type of a species will be alive on this earth because of reproduction. Because every organism arise from these similar organisms only. The lions can't form dogs. Dogs only can form dogs. Lions can give birth to lions only, not the other one. So lions will be alive. This particular type of lions will be alive only when that particular type of lion reproduces. Okay, and second significance?
It is important for the whole ecosystem as the to balance the nature.
Okay, so for balancing it, maintaining the ecological balance. Also to preserve a particular type of DNA. So why there is a problem to this one? That's extinction. Suppose some of the species are endangered. They have marked endangered. Why there is a problem?
Because their numbers are low due to poaching or bad environmental conditions.
And
they become extinct.
Okay, then fine. Then what will happen?
Ma'am, there will be a break in the food chain.
No, there are food web is working. Food chain will okay. If food chain is not working, food web will work. So we will lose a particular type of DNA. So because the Asiatic lions are there, so Asiatic, tigers are there. So those tigers are give rise to the similar type of species, cannot be formed. And if they get extincted, we will not get the particular type of DNA to reform. Right? Plants are there. If a particular plant is undergoing extinction, what will happen? If we have DNA, we are able to produce the similar type of or this plant. But if that any of the part is not available, then that DNA will not be available. Then the reproduction could not be possible. So that is okay, important fact, right? Then what is the importance of DNA copying? 1 mark question, what you will write? Give specific answer. It is a 1 mark question, so it is necessary to give a specific answer.
Do we write it?
Yes, if you want, you have to write down. These are important questions I'm dealing with. So that is which are they have asked in earlier exams.
So we should write the questions.
Yeah, better.
Okay.
Ma'am. Ma'am.
Yes.
Ma'am, for the importance of DNA, is it for the inheritance of particular characteristics?
DNA copying, importance of DNA copying. So what happens due to DNA copying? Yes,
Ma'am,
your answer is right.
each cells have many cells have different functions, so there can't be any common DNA for all the cells. So DNA copying is essential for each cell.
Simple question, simple answer. We are not going in much detail for that. DNA copying means what? The one cell's DNA has copied, and for give that DNA cannot survive alone. So DNA copying has to be taken place. After that, if that DNA comes out of the cell, so it has to show its properties. Now it has carried information on itself, right? It has a genes. So it forms a genes. Now, if that copying has been done, so copying is importance of that copying. What happens by that copying? The characteristics from parent cell will be transferred to new cell.
Ma'am, I have a question.
First, listen properly. What I'm telling. What is the importance of DNA copying? So, what is the immediate effect due to DNA copying?
Variation.
No, I'm asking immediate effect. Due to DNA copying, what happens immediately? What change has been taken place immediately?
New cell is formed.
And that will, this one, and then that cell will have?
Cell apparatus.
That is a cell is a cell apparatus.
Ma'am, same properties as the parent.
Same properties are transferred, no. So by DNA copying, DNA is what? It is an equation of the earlier cell, which is transferred to the new cell. So what is happening? All the characteristics from the first cell, it is transferred to the second cell. So it is helpful in transferring similar characteristics from parents to offspring. Immediately by DNA copying, what happens immediately? That is a transfer of characters. DNA carries information. So that information is transferred to the new cell, and new cell will start behaving like an old cell because it has exact similar structures. Because the similar information is transferred to it. So it will behave exactly similar. Right?
Yes, ma'am.
What about genetic variation?
Variation because of the DNA error, not by DNA copying. In DNA copying, if there is some percentage chance are there, if it is while copying, there is a mistake happens. So while I am dictating, sir has dictated you and you have written something else, what will happen? Meaning of that line will remain same?
No, ma'am.
The information is changed.
Yes, ma'am.
So sir wants to convey something else and you have taken something else, you have written something else. So therefore you are considering it as a something else and you are transferring the same information to different person. So due to that, what happened? Every person has understood the concepts differently. So that is what a variation.
Okay, ma'am.
Okay, you might have seen one video. That is, one person has written on something on back and that is transferred one by one to the different people. So finally, what happens? What was told and what is transferred to the 50th person?
Ma'am, it's completely different.
It is completely different. That is what a variation is.
Okay, ma'am.
Clear? So message which is passed on has some changes, some errors. So due to that error, the meaning of that particular function has completely changed. So that is the DNA copying error, and that causes variation. That leads to variation. Clear? And many variations leads to evolution. Clear?
Yes, ma'am.
Yes, ma'am.
But DNA copying won't ensure variation. It only ensures the transfer of characteristics from parent to offspring or parents to progeny. And in that copying, if there is some change has taken place, DNA when they combines with the chemicals have combined. So that chemicals have not combined, one of the chemical has not connected properly, then there is an error caused. And that error causes variation, changes. Clear?
Yes.
Now, the different types of reproductions. Types of reproduction,
Asexual and sexual.
differences, asexual and sexual reproduction. So here and this one, what are the differences, main differences between these two?
Multiple organisms are formed in asexual.
Okay, first one important is only one parent involved. And your sexual reproduction, two parents involved.
More variations are created in sexual than asexual.
That is the last point. Here, no gamete cells required, no specialized cells, not gametes, specialized cells. That is gametes required. And here, gametes are required. Specialized cells called gametes required.
Characteristics of only single parent variate.
Ma'am, fertilization does not occur, fertilization occurs.
Fertilization won't take place. Fertilization takes place.
Characteristics
No or less variations. More variations. Due to combination of DNAs of two parents. Exactly similar to parent and similar to both the parents. Exactly. Characteristics exactly similar to parent. That is less variations. And here characteristics similar to both the parents. Okay. Then the types of asexual reproduction. First one is fusion. First one is fusion. Second one is fragmentation. Third one is budding. Fourth one is regeneration. Fifth one is spore formation. And sixth one is vegetative propagation.
Ma'am, regeneration is not a mode of asexual reproduction, right?
But it has to be considered according to NCERT, you have to consider. So in binary fusion, you have to remember the examples of, so first is fusion. In that binary and multiple fusion. Binary fusion, you have to remember the two examples, amoeba and leishmania. What is the difference in amoeba and leishmania? Binary fusion. Both have undergoes a binary fusion, but there is some difference. What is it?
Ma'am, in amoeba, it can occur in any axis or plane, but in leishmania, it is only in one plane, which passes from the mouth to the tail.
That is flagella, we call it as
Flagellum.
flagellum. Okay, amoeba has a irregular shaped body. So it the fusion takes place through any plane, through any axis. There is no specific axis for fission. But in Leishmania has a one specialized part that is called flagellum. So the flagellum should divide in both the it should enter both the cells. So therefore, in Leishmania, fission takes place through only one plane or one axis which passes through flagellum. And you need to know the diagram of both. They have asked this question for 3 marks. So amoeba and Leishmania both undergo binary fission, but still the binary fission in both of them, how the binary fission in both of them is different, explain with the help of diagram. So you need to know both the diagrams.
Ma'am, could you explain again?
Amoeba reproduction, this binary fission, that is a dividing cell into two different cells, two daughter cells, will take place through any axis. But in Leishmania, it always taken place. The fission takes place through a special one single axis which is passing through flagellum. And turn on camera, everyone. How many times should I tell that? Okay. So amoeba through any axis, Leishmania fission through single axis. And multiple fission. Only one example you have to remember, that is Plasmodium, malaria causing protozoa. Plasmodium is a protozoa causing malaria. So Plasmodium first at the time of under maturity. At the time of maturity, Plasmodium forms a cyst, and inside the cyst, the nucleus divides multiple times. And how many times the nucleus divides, that many times the cytoplasm divides. And then finally, many cells, many plasmodia are formed inside the cyst. And once the complete division takes place, the cyst breaks open and all the plasmodium will be free to live individually. So Plasmodium multiple fission. Multiple fission, that means fission in multiple times. One single nucleus divides multiple times. So here, single plasmodium, then nucleus divides multiple times. So one cyst is formed above it, and then that many times the cytoplasm also divides. So that many cytoplasm, then once the full cytoplasm divided, cell membrane is formed around every plasmodium. Then this cyst breaks open, ruptures, and then these plasmodium will burst open and start living as an individual plasmodium. This is multiple fission.
Ma'am, can we draw this?
Not needed.
Okay.
Right now, not needed because you already have done that. You have to do that one. You need to remember that. In case of Leishmania and amoeba, first it will absorb, intake more cytoplasm. So first step is absorbs more cytoplasm. At maturity, it will needs more. So more cytoplasm will be formed. After that, when it will start this one individually, the nucleus starts dividing. First, the nucleus divides, and then through the flagellum will start dividing. and then the full body. Like this, there is a constriction is formed in the cell membrane. This is a cell membrane. So constriction will be formed. So in that constriction, the flagellum also first divides, and then through this plane only, there are two flagellum will be formed, start outgrowing, and then the constriction is formed, which divides the cell membrane into two parts, having nucleus in each, and then through this plane, the leishmania completely divides and separates, start living into individual. And one more thing, remember in fission, the parent identity has lost. Fission is only one type of reproduction in which parent identity has lost. So after forming new individuals, there is no parent left out. Right?
Yes, ma'am.
So
Yes.
objective question can be formed here. This one, the type of asexual reproduction in which parent has lost its identity.
Ma'am, is it the same in multiple fission?
Yes, both. The fission only.
Ma'am, so in plasmodium also the other cell dies after giving birth to so many.
Cell dies is not, nothing will die. It is divided.
Okay, cells is okay, ma'am.
The single cell is only divided into and form into new.
Ma'am, in regeneration also parent identity is lost, right?
So it was already breaking itself own. It itself breaking its body. Then next is fragmentation. So fragmentation example you have to remember is
Spirogyra.
Spirogyra. So what happens in that?
In
What happens?
Spirogyra, the body breaks itself into small fragments, and each of that fragment is able to regenerate the rest of the parts of the body.
So there is a Spirogyra. So first, this is a one filament. It is a filamental body. And this will take place in a cellular level of body organization, not in tissue level of body organization. Fragmentation and regeneration is somewhat similar. But in what way they are different? Fragmentation takes place in very primitive organisms. The filamental body, whereas the body organization is cellular. So the cells are not divided further to form tissues. So in such organisms, the fragmentation takes place. Spirogyra, cellular level of body organization. So these are cells have been connected together in the form of filament. So these filaments break into multiple. And these every filament will again grows further to form the individual new Spirogyra.
Ma'am, in Plasmodium, the parent cell divides into a lot of parts because of the unfavorable conditions, right?
Yeah, under unfavorable conditions, or they want to divide when they have favorable conditions. Under favorable conditions, they will divide.
Oh, okay. So when do they break?
When they are inside our blood, or when they are inside female Anopheles mosquito's body, that time.
Okay, ma'am. And
Otherwise, they can't. Otherwise, they can't.
And they even break their cyst in favorable conditions.
Yeah.
Okay, ma'am.
It's clear here. So what is the difference? This is the similar case you can see in regeneration also. So what is regeneration? Next step is regeneration. So regeneration is also the same. Then in which way they are different?
Ma'am,
Yes.
Ma'am, in fragmentation, the parent cell divides in itself into small parts, and then from there, the rest of the part grows. But in a fission, I mean, the parent cell just divides itself, and I don't know how to explain.
Question asked for 2 marks.
Ma'am, so how are they different?
That's what I'm asking you. How are they
Ma'am,
different?
I guess in fragmentation, it divides, and the parent
Yeah, actually, I
dies.
told you your answer also. The processes look same exactly, right?
Yes, ma'am.
But in which way they are different?
Ma'am, fragmentation occurs in organisms that have cellular structure, but
No,
in tissue-based organisms, it is regeneration.
regeneration, because
And
specialized tissues
regeneration
are.
occurs due to accident. In fragmentation, the body itself breaks down purposefully.
Yeah. No, that happens in this one also. Regeneration also that happens. Only main difference is the cellular level and tissue level of body organization. The second one is the specialized for regeneration, specialized tissues are needed. For forming the new body, specialized tissues are needed to reform the new body, the lost part. But in case of fragmentation, it's not needed. It is a cellular level of body organization. Different normal cells are only need to be formed. There is again, those cells are not combined to form any tissues. So therefore, there is no specialized tissues required. Specialized cells are not required for fragmentation and the regrowth. But in case of regeneration, there are specialized tissues are necessary to reform, regrow the lost body part. These are the main two differences between regeneration and fragmentation. Understood?
Ma'am, so fragmentation, you don't need a special tissue for it.
Special tissues are not necessary
Okay, ma'am.
and takes place in cellular level of body organization. And regeneration, specific cells need to divide to form the lost body parts and takes place in tissue level of body organization. The organism which have tissue level or body, this one organ level of body organizations. So these are the main 2 points of difference.
Ma'am, can regeneration be considered a mode of fragmentation?
Fragmentation, that's what I told you. Fragmentation don't need any specialized tissue because their body is very simple. Only cells need to be formed. And in case of here, the tissues has to be formed, and that tissues can be formed. The new type of tissues can be formed from a special cells. Not all the cells could not form the different types of tissues. Right?
Yes, ma'am.
So here, in case of regeneration, you have to remember the diagram of planaria. And even Hydra also. Hydra also divides by regeneration, and cut body parts, lost body parts will regrow again with the help of specialized tissues. Clear? Then the third one is budding. Next one, fourth one is budding. Budding in Hydra. Always ask question for 3 marks. Explain budding in Hydra with the help of diagram. Budding in Hydra is asked, and for practical purpose, they are asking budding in East. Okay. So budding, what happens in budding? How you will explain the process of budding? Budding, that means?
A small bud-like structure is formed on the membrane of the organism. That small bud grows to form a new organism, and then it detaches from its parent to
Yeah.
form a new organism.
Yes. So word when it is grows. So word use proliferate. The word used there instead of grows. The new cells will grow on the body of the organism. The word used is proliferate. Grows intensively is proliferate. right? And budding in hydra. The diagram of hydra budding is important, and they asked multiple times. Under favorable conditions, small bud like outgrowth is appears on one of the end of the hydra parent cell, and those cells gradually enlarge in size, proliferates. That means here multiple cells, intensive cell formation takes place at that place. The nucleus enlarges and belongs to this one. The new small hydra start growing on the parent body. When the full new hydra matures, then it detaches itself from the parent body and start living as a new individual. Okay. Understood?
Yes, ma'am.
So 3-4 lines only you have to write. Draw the diagram and 3-4 lines you have to write. Step by step drawing diagram is important, and 3-4 lines that I notes I will give you. That's you have to write from that. Okay, it is given in NCERT. Not much. One line only they have mentioned in NCERT. Then after that, the spore formation. Spore formation takes place in
Fungi.
Yeast, ma'am.
fungus. Fungi mostly. Remember, you have to remember one name. Rhizopus. That is bread mold. The parts of rhizopus.
Ma'am, it forms small microscopic spores and then it bursts out.
So what are the parts of it? This stem-like structure is called as hyphae, and this balloon-like structures,
Sporangium.
sporangium. And inside sporangium, seed-like tiny structures are present, are called as spores. Okay, which are reproductive material. Okay, so hyphae, and then below that the root-like structures are called rhizoids. The root-like structures called rhizoids. Over that stem-like structure called hyphae. Over the stem-like structures, there are the balloon-like sporangium are present, sporangia, plural. And that sporangium contains multiple spores. Tiny spores are present. They are very lightweight. Now, spore formation, advantage of spore formation. Question asked, that is, what is the advantage of spore formation? What can you write?
Many offsprings can
easy
be
to
produced so
disperse.
that their survival can be ensured.
Okay.
Ma'am, easy to disperse.
Easy to disperse, they are very lightweight, so can be easily carried out through wind, air. Until maturity, they are protected under sporangium. So till maturity of spores, they are protected under sporangium. After maturity, sporangium burst open, and those spores can be easily carried out through air also. And then under favorable conditions, they sit on the organism and then start regrowing. So now what are the necessary conditions for growth, spore
Moisture.
growth of the individual from the spores?
Warmth, humid environment, then
Humid
availability
environment.
of food.
Okay. and warm temperature. Moisture, warm temperature, and dead matter. Clear? So these are the questions asked for 1-2 marks, always asked. What are the advantage of spore formation? And second one is, what are the favorable conditions for the spores to grow? So the important conditions are moisture, warm temperature, that is a humid weather. Altogether it is a humid weather only, and the presence of dead decay matter. Clear?
Yes, ma'am.
Then next one is vegetative propagation. So what do you mean by vegetative propagation?
Part of a plant are cut and sown in the soil so that they can grow to form a plant.
Reproduction through vegetative parts of the plant. So what are the vegetative parts of the plants?
Stem, root, leaf.
Stem.
Except flower, all other parts. Roots, stem, leaf. Okay. So these are called vegetative parts. And if the reproduction takes place through any of these parts, then it is called as vegetative propagation. Okay. So through that, how the bryophyllum reproduces? Which vegetative part of the bryophyllum helps in reproduction?
Leaf.
Leaf. Bryophyllum reproduces through leaves. Ginger?
Stem. Underground stem.
Underground stem. Banana?
And
Banana?
that's artificial vegetative propagation. It doesn't contain seeds.
So why artificial? It is through stem also that happens.
Ma'am, is banana
Cutting.
stem?
Cutting is used for banana. Jasmine?
Stem cutting.
Stem cutting or layering also we can use. Stem cutting, layering. Rose?
Stem cutting and layering.
Not layering. Stem
Not layering.
cutting.
Okay.
It's only stem cutting because it won't have the this way, no, vine-like structures. Jasmine has vine-like
We can
structures.
use grafting also.
Rose grafting, yes, can be done. Cutting,
Okay.
cutting and grafting possible. Layering is for jasmine.
So,
And money
ma'am,
plant.
the plants which have vine-like structures
Yeah, those can be
can be
grown
used in layering.
Layering, yes.
And nearly
Because
all
what is
plants can be
layering?
used for grafting.
Yes. So now here, what is layering? If it has a vine, it is moving like this. If it has a vine-like structure, it will bend downwards. And then we are immersing it inside the soil. Now here, when that part get immersed in a soil, so what happens here? The roots will start forming. Here the roots formation takes place. And due to that, if you cut this branch, then also here the new individual grow from this. That we do in money plants also. Cutting, that means stem we cut and then we put it in a soil, watering it, nutrients if it gets, it will start growing from that. Hibiscus can be done by stem cutting. It undergoes sexual reproduction, but also with the asexual reproduction by cutting, that will work. Okay. Now, what are the advantages and disadvantages of vegetative propagation? Always asked question, 101%. That question will be there. The advantages of vegetative propagation.
Faster growth.
Genetically similar.
One thing, keep in mind, if you are writing a very general answer, how you will make your paper more impressive?
Ma'am, write in point.
Give headings
How
and
you
points.
picking more impressive? You want to look more impressive, what you do?
Underline main words.
Okay, and
Make
now
headings.
like this, so you want to look more impressive, what you do? Especially girls, what you would do?
Underline.
Underline.
Highlighters.
Looking wise, I'm telling.
Highlighters.
No, we don't use highlighters. You should not use highlighters in answer sheets.
Use
Suppose
points.
we want, in general case, when we want to look special, different, what we do, we generally choose the things extraordinary, which is not ordinary. Dresses we select, which are not common. We generally select the things which are completely uncommon, which is not wearing. So it is a new one that we wear. The same thing when you are writing answer, something should be, there is a uniqueness should be there in your answer. So if you write all the in general answers only, so that is a common answers if you write, so will it become more impressive? It will look like a general case only, right? General answer only. So you have to add some of the places, it is possibility, not every time it is not possible, but some of the places it is possible. So that uniqueness, you have to show your uniqueness, unique quality you have to show. Just like in this question, everyone will write, it is a faster growth, genetically exactly similar, that will come. So those are the common points. You want to write a specific point, what you will write? What you will write a specific point for disadvantages. Then listen. If you don't know, write it down. Sir has told that I've listened at that time also. The plants which could not produce viable seeds can be reproduced by vegetative propagation.
Can you repeat it?
Plants which could not produce viable seeds. Use the word viable always. Plants which do not produce viable seeds can reproduce vegetatively, just like a banana. Banana produce seeds, but those are not viable. Viable, that means those could not reproduce. Those seeds could not
developed,
reproduce
not
further.
mature.
Could not mature, could not germinate. They are not able to reproduce into new individual. So such seeds are called viable. Which one? Not viable seeds. Clear? Viable, that means which can reproduce, and non-viable means which could not reproduce further. So those seeds are useless. So if the plants is able to produce non-viable seeds, they can be reproduced by vegetatively. Just like for example, is a banana, main common example. And the second one, this is a first important point you have to mention. And the second one, what was that? Yeah, these plants bear fruits and flowers much earlier than sexually reproducing organisms or sexually reproducing plants. Plants which reproduce vegetatively. These plants means what? Plants produced by, formed by, or grown by, sorry, grown vegetatively, forms or bear fruits, flowers much earlier than sexually reproducing plants. And then you can add all common properties, advantages. Faster growth, and again, more number of plants can be reproduced. More number of plants are produced.
Ma'am, exotic plants can be another reason.
Yes, exotic plants can be reproduced by this, right? And then genetically similar. Genetically similar to parent plant. This is actually a good thing, advantage also, and the same thing will become a disadvantage also. So less variations.
Isn't the first point of the isn't the faster growth and the second point
Second point are
same?
same. Yes, they are same.
So, ma'am,
you can write more number of
second-
plants are produced in shorter duration of a time.
Okay, ma'am.
Are produced in shorter duration. And genetically similar plant. Now, what are the disadvantages? Question is asked, write 2 advantages and write 2 disadvantages.
Disease can be easily transmitted from the parent plant to the offspring plant.
Yes. So here there are very less variations and genetically similar. So if genetically similar is good characteristics are going, then possibility that is a bad characteristics are also passing. That is, a diseases also can be easily transferred.
And another disadvantage is that there will be no variations.
There is no variation, or very less or no variations.
The species won't be able to survive if sudden change.
Survival advantage will not be there. Variations gives survival advantages, but due to asexual reproduction, there are no variations. So survival chance is less compared to sexually reproducing organisms. Understood?
Yes, ma'am.
So now if here the question is asked about tissue culture, although tissue culture they have explained in one box. But if school has asked question based on tissue culture, that is advantages of tissue culture, you have to mention all these advantages of vegetative propagation for tissue culture, nothing else. Clear? So vegetative propagation advantages and tissue culture advantages are exactly same, no difference.
Ma'am, so the disadvantage will be also same?
Same, yes. Everything, because the tissue culture is a part of vegetative propagation only, right?
Yes, ma'am.
So it will remain same. This is a one part of the chapter. Now, second part of the chapter, that is sexual reproduction. So in that sexual reproduction, first you have to learn about the plants and then about humans. So in plants, first you have to remember longitudinal section of flower diagram, 4 whorls. They can ask you 1 or 2 marks questions on based on this. Which are the male reproductive parts of the plant? Flower. Male reproductive part of the flower. What is that?
Stamen, anther.
Stamen and anther. Stamen consists of filament and anther, no?
Yes, I'm sorry.
Stamen is the male reproductive part. It contains 2 parts, filament, over that anthers. Inside anthers, there are pollen grains. Inside pollen grains, pollens are present. Inside pollens, there are gametes are present, male gametes. Clear? And female reproductive parts in flower?
Stigma, style, ovary.
Stigma, style, and ovary.
Ma'am, or if they ask, could we write pistil
The full name is called pistil.
and just leave it there? Like, what is the female part? Pistil. Leave
Yeah,
it.
pistil.
Okay, ma'am.
Ma'am, I have a doubt.
Yeah.
Ma'am, you told flower diagram will come. So will it be a
Yes.
bisexual flower or unisexual flower?
Bisexual.
So no unisexual flower diagram?
No.
Okay, ma'am. Okay, ma'am.
So here, both of them, this is the 5 anthers you can see. This is the stamen part. So filament, stamen consists of filament and these 2 anthers. Inside anthers, pollens and male gametes. This middle part, bulge part, is a female reproductive part. Upper part, funnel-like structure, sticky, that is stigma. Then middle tube-like structure is called style, and the lower bulged balloon-like structure is ovary. Inside ovary, ovule is present. Right? So this diagram you have to draw when the question is asked to draw longitudinal section of flower. And then what is this one? Types of flowers?
Unisexual, bisexual.
Examples?
For both of them, hibiscus.
Both of them hibiscus, no. Hibiscus is only bisexual.
Bisexual, China rose, unisexual.
Bisexual is hibiscus, that we can write. And any other? Jasmine?
Sunflower.
Mango, sunflower, all these are bisexual flowers. Only thing, all gourd families are unisexual. Bitter gourd, bottle gourd, ridge gourd, cucumber, papaya, these are all unisexual. Watermelon, these are all unisexual. Okay, unisexual, that means they have either male reproductive system or female reproductive system, any one in one flower. That is called unisexual. And bisexual means they have both in one single plant. So you can see the diagram here, that is a bisexual flower. So they have their own, this one also, what do you call, male reproductive parts as well as female reproductive part in a one single flower. That is bisexual. Unisexual, only one, either anthers or either carpel, pistil. Clear? Examples of unisexual you have to remember: papaya, cucumber, at least 2 you can remember, watermelon. These 3 you can remember. Or unisexual flower.
For bisexual, we can use hibiscus and
Hibiscus, rose, or mango. Okay. Then next is fertilization in flower, in bisexual flower. It is first step is pollination process. So pollination important. What is pollination and what are the 2 types of pollination? Question always asked. So what is pollination means?
And the
Process of transferring pollen grains from the anther to the stigma to perform fertilization.
So here the pollination is the process of transfer of pollens from anther to stigma. Is called as pollination. 2 types of pollination: self-pollination and cross-pollination. Self-pollination means same flowers' pollens are transferred from anthers to stigma of the same flower. Remember, there is a confusion between this. Teacher also created a confusion. Single flower. The gametes, the male gametes of that flower will be transferred to the female gametes, means female part of the same flower. Then it is called as self-pollination. And if the same plant's second flower, from that, if the pollens are transferred to the second flower, or from any other plant, if the pollens are transferred to different flower, then it is a cross-pollination. Able to understand?
Yes, ma'am.
Only one single flower, if the gametes, pollens are transferred from that one flower to the same flower's stigma, then it is called self. From the same plant's second flower, if the transfer is taken place to the third flower, then that is cross-pollination. Understood?
Yes, ma'am. Ma'am, for both the methods, you require a medium, right? Like insect, wind.
Self, generally not needed. You can, it will be useful, but if it is not, then also okay.
Ma'am, can we say?
Yes.
Ma'am, can we say that unisexual flowers usually perform self-pollination?
Pollinating agents. Without pollinating agent, unisexual flowers cannot reproduce. A fertilization won't take place.
No, sorry, bisexual plants. Do they usually?
Yes.
Do bisexual plants usually perform self-pollination?
Not necessarily. Not necessarily. Depends. Now in this flower, self-pollination not possible. It's easily possible, but not without pollinating agent, not possible. Because stigma is at the higher than the pollens. So to jump them will be much more difficult. So much more possibility is only by cross-pollination. In some plants where the self-pollination is happening every time, in that case, the stigma size, height of the stigma is less, means that the carpel height is less. So immediately this one, pollens will immediately fall down.
Ma'am, and what is double fertilization?
That part I have not started anything about the fertilization.
Okay, ma'am.
So here,
just a small doubt.
Yeah.
So unisexual flowers usually go for self-pollination.
No, they won't go.
Achha, that is cross.
That is always cross because they don't have their own female and male gametes, both. So they have to follow the cross-pollination process.
Okay, ma'am.
And cross-pollination agents are needed. Without pollinating agents, the pollination won't happen. Pollination takes place with the help of pollinating agents. Self-pollination in some of the plants, if conditions matters, that is if the size of this carpel is smaller than the anthers, then obviously the self-pollination can be done easily. So first step of the fertilization is pollination. By the pollination process, male gametes are transferred to the female reproductive part. Stigma is sticky, so immediately pollens will stick to the stigma. You can see here diagram. This is a funnel-shaped structure which we call as a stigma. Stigma is sticky. It keeps secreting the sticky chemical. So due to which the pollens easily stick to the stigma. And inside that, there are actually two types of cells are present inside the pollen. One is a male gamete, two male gametes are there, and the tube cells. So those tube cells will start forming a tube, pollen tube. So therefore it is called tube cells. So tube cells keep forming this pollen downwards and moving downwards here till ovary, the tube cells will form. At the end, tube cells will burst open and the male gametes which travels through this tube is called as pollen tube. So these male gametes will move towards ovary and they will open into ovule. Inside ovule, there are female gametes. So one of the male gamete fuses with this female gamete and the fertilization takes place. Fertilization is the process of fusion of male gamete with female gamete and forms one single cell called zygote. So fertilization is a process of fusion of male gamete and female gamete to form zygote. So here one of the male gamete enters ovule, fuses with female germ cell or female gamete and forms zygote. That is one fertilization. And the second one is second male gamete also sometimes enters, and there at the center you are able to see two cells. So those are called polar nuclei. So one of the second male gamete fuses with this two nucleus and fertilization. Now three cells combine together to form one single cell. That is called as triple fusion. And therefore this is one fertilization of triple fusion, one fertilization. And here zygote formation is one fertilization. So together it is called double fertilization. Able to understand process of fertilization?
Ma'am, I didn't get you. Could you repeat?
When pollens stick to the stigma, then inside that one specialized cells are present, they will start forming tube. That is called as pollen tube. So that pollen tube will arise from stigma to ovule. So through that pollen tube, male gametes will be carried towards ovule. One of the male gamete fuses with female gamete and forms zygote. That is one main fertilization. Now sometimes the second male gamete fuses with two cells at the center. So three cells combine together. That is also a fertilization. So that type of fertilization is called triple fusion. Because three cells, these are polar nuclei. These are at the center, there are polar nuclei. These fuses with one male gamete. So this way, three cells combine to form a one cell. So this is called triple fusion. And there is fusion takes place in this one. And as well as here, male and female gametes combine to form a zygote. This is one fertilization, main fertilization. So this one and this one combines, forms double fertilization, and that is only what we call as double fertilization.
Ma'am,
Yes.
and what are polar nuclei?
There are the cells. There are two cells present inside the ovule, other than female germ cells. They always stick together. They always remain together.
Got it.
Got it, ma'am.
So that is double fusion. Now this is what the fertilization. After fertilization, this zygote divides further and form embryo. Okay. So zygote divide further to form embryo. So that embryo will remain inside ovule. And now to protect that embryo, the changes will take place in this ovary. So those changes are called post-fertilization changes. So in these post-fertilization changes, what are the changes takes place? So zygote forms embryo. A cell, zygote divide further, form the multiple cells. So those multiple cells combine and form embryo. Now embryo is present inside ovule. So ovule has to protect it. So ovule gets converted into seed. So ovule gets hardens and converted into seed. And then ovary also has to protect seed. So ovary gets converted into fruit. And other parts of the plant fall off. A flower fall off. Zygote forms embryo, ovule gets converted into seed, and ovary gets converted into fruit. And remaining parts will fall off. Just like a style stigma goes, sepals, petals fall off. Sometimes sepals remain with the plant, remain with the fruit. Otherwise, it is most of the time it is fall off. Understood? These changes you have to remember. Zygote converted into embryo, ovule converted into seed, and ovary converts into fruit. Clear?
Yes, ma'am.
Yes, ma'am.
So these are the post-fertilization changes. Then the structure of seed also you need to know. The parts of the seeds. So 3 parts you have to remember. What are 3 parts of seed? And this diagram, they have asked this diagram for case study question. Pollen tube formation. Whenever the question is asked to explain fertilization in plant, you have to explain this diagram. This diagram you have to draw. So seeds part.
2 cotyledons and 1 baby plant inside.
Some are more.
Is it radical or something? Radical and plumule.
Plumule and radical.
Yes.
So it is the part, there is inside that embryo is present. So when this seed has a outer coating, the structure, 2 parts are there. That this is outer part is a seed coat. Inside that 2 leaf-like structures are present. So those are called as cotyledon. When you open the outer covering, when you open a, suppose this one is there, gram is there, or pea. So when a pea is green outside, so that outside there is a 1 layer. So that is seed coat. Now you open a layer, then if you put it in them into 2 parts. So those 2 parts are called as cotyledons. So inside cotyledons, there are 2 again structures are present, which is whenever that plant germinate, whenever the sprouts, have you seen sprouts?
Yes,
so one white part, when you sprouts means it is get germinated, 1 white part start coming out of it first. So which will move in upward direction. So the part of the plant which moves in upward direction, that is called as plumule. This diagram is wrong. It's not contain all the parts. So that part is called as plumule. Plumule is future shoot. Plumule is a future shoot. And one more part will come, that is radicle, which is a future root. Able to understand?
Yes, ma'am.
Ma'am, can you repeat?
Plumule is a future shoot. And a radicle is a future root. Just a sec, the diagram NCRT don't have the full.
Ma'am, tomorrow I won't be attending the class. Day after tomorrow I have exam.
Yes, ma'am, me too.
Plumule is not sure. Epicotyl and dicotyl only they have told it. So plumule, remember plumule is a future shoot and the radicle is a future root. So one part which moves downwards, that is a radicle, and one part which moves upward, that is a plumule. Plumule gets converted into shoot. It will start forming a stem and shoot structures. and the radicle will grow downwards and form root structure. And the cotyledon will form the first leaf. Cotyledon will form first leaf, will become first leaf when the plant starts growing. Okay. So that you have to remember. Okay. So these are the parts of this one. And whenever the question is asked about the fertilization, you have to draw this pollen tube diagram. This pollen tube diagram is very important when the question is asked to draw, explain fertilization in plants. Clear?
Yes, ma'am, clear.
So till this part, any doubt?
No, ma'am.
Everything clear? Now the next part is, I'm not taking today maths, we will take tomorrow full maths. Okay. Now next is reproduction in humans. In that male reproductive part and female reproductive part. Diagram important, and even the different parts and their functions are important. Different parts of the reproductive system is important.
Ma'am, can you tell the function of prostate gland?
Yes. Prostate and seminal vesicle both together, you have to remember.
Ma'am, what is the
Not
function?
separately, they have not given. One single function only they have given. So that is two functions they perform. One is, so everything I will explain. Wait, give me a second. So in this case, there are these two structures called testicles. Inside that, testes are present. So first important point from a part, that is testes. So testes function. There are two functions testes possess. So one is, what is the first function? Important function of testes. It is endocrine and exocrine too. Yes.
Produce testosterone which regulates the secondary sexual characteristics.
Not able to hear you properly.
Ma'am, they produce testosterone.
They secrete testosterone hormone. So first function of testes is to secrete testosterone hormone. First is secretion of testosterone. And why it is testosterone important? It helps in testosterone, which helps in development of secondary sexual characters. And the second function is to form male gametes, that is sperms. Right? These are the two main functions of testes. Next one is vas deferens. So vas deferens is tube-like structure which carries sperms from testes to urethra. So vas deferens's function is to carry sperms. It is a tube-like structure which carries sperm from testes to urethra. Before that, one more function part is there, that is called scrotum. Scrotum is also called temperature regulator, a thermoregulator. So thermoregulator works as a temperature regulator. So these testes are present in a sac called as scrotum. So the function of scrotum is a temperature regulation of sperms. Sperms will survive at a temperature 1 to 3 degrees Celsius lesser than body temperature. Sperms can survive to a temperature lesser than 1 to 3 degrees Celsius lesser than body temperature. So scrotum is providing that specific temperature. Clear? Everyone able to understand? Case study question is complete possible on this part. Okay. So scrotum, it is not given in your textbook, but yes, they have asked. Vas deferens clear? Then next is seminal vesicle and prostate glands. There are glands are there. There are 3 types of glands, but they have not mentioned in your textbook. That is only 2 they have mentioned. One is prostate gland. And seminal vesicle. So what is the function of these 2 glands? These 2 glands secretes. Glands' function is to secrete. So these 2 glands secretes. What they secretes? They secrete a fluid. They provide the fluid medium for easy transportation of sperms. They provide medium for easy transportation of sperms. So fluid medium, because sperms are cells, they have to move from one place to other place. So they can swim, sperms can swim. So they need a fluid medium. So that fluid medium is provided by these glands. And second function is to provide nourishment to the sperms. Sperm has to survive 4 to 5 days outside body. So sperms survives 4 to 5 days outside male body. So for that thing, they need nutrition. So that nutrition is provided by these glands. Clear, Ashish? 2 functions?
Yes, ma'am.
So providing fluid medium for easy transport of.
Ma'am, is there any name for the
Semen.
Okay.
And then another function is to provide food, nutrition to sperm. So generally fructose is added. And that medium is alkaline medium. Okay, so always ask question for 2 marks, write the functions of prostate gland and seminal vesicle. And then urethra, it is a common opening for both urine and sperm. So common path for discharge of urine and sperm is urethra. Okay, so these are the parts of male reproductive system. Understood, everyone?
Could you repeat what is semen? The fluid medium.
Semen is the fluid, sperm along with the fluid medium with nutrition. Sperm plus fluid medium plus nutrition together is called a semen. They have asked this question. Let me show you. 2020 board paper they have asked. So I'll show you the question asked on this part. Male reproductive system, they have asked the question for 5 marks. I got it. Level parts A, B, C, D. So what is A? Yes.
Urethra. Urinary bladder. Urinary bladder.
Seminal vessel.
Where is it? Is its entry point? Where is its entry point?
Ureter.
Huh? Couldn't get, come again?
Ureter.
Yes, A is ureter. It is a bladder. Which tube collects this one, brings the urine to bladder?
Ureter.
Ureter. So A is ureter. B is this part, no, just below this one, urethra. Oh, sorry, urinary bladder. These are glands are there. These small glands are present. So those are seminal vesicle and prostate glands. Prostate glands, you can write it as. Okay, B is prostate gland. C is
Ma'am, C is
Urethra.
no.
C is
Ma'am, is it
Ma'am, it is
sperm
urethra.
duct?
Ma'am, urethra.
so it is, this is the path you can check here. So the bladder output is connecting here as well as this in output is also connected here. So both of them connected together to C. So what is C?
Urethra.
Urethra. And D is?
D is sperm duct. Vas deferens.
Vas deferens. Sperm duct or vas deferens.
Ma'am, can we write as sperm duct instead of vas deferens?
Yes, you can write, but better vas deferens if you remember.
Okay, ma'am.
Name the hormone secreted by testes and mention its role.
Testosterone.
Testosterone and its role is?
Developing secondary sexual characteristics.
Yes, developing secondary sexual characters in male. State the functions of B and C in the process of reproduction. So B is prostate gland or seminal vesicle. So both the functions you can write. That is?
Provide fluid medium for easy transportation of sperm.
Yes.
Also provide nutrition for sperm.
Yes. Providing nutrition and this is providing fluid medium. Okay. So the next one, then the next question. And C is a function of C?
To
transport pathway for
to
semen and urine.
Passing the common passage for the discharge of urine as well as sperms or semen. Okay, understood? So such questions can be there. Then next, female reproductive system. The parts of female reproductive system. Diagram important, always ask diagram for 3 marks. And they will ask you questions, label the parts explained below. So parts explained below, that is the place where eggs are formed. Label the part where the eggs are formed.
Ovary.
Ovary. The part where fertilization takes place.
Fallopian tube.
Fallopian tube. And the place where growth of the fetus takes place.
Uterus.
or embryo implants, or the place where embryo implantation takes place? Uterus. Okay. So parts of this female reproductive system is 2 ovaries on either side of uterus. Almond shaped ovaries are there, in which eggs, female gametes, that is eggs, ovum, ova are formed. Eggs, ova, both are same. Ovum is singular, ova is plural. So they are formed in the ovary. Every ovary, and after maturity, one of the ovary releases eggs in this tube called fallopian tube. So once in 28 days, each ovary alternately releases eggs in the fallopian tube. That process is called ovulation. The release of ovum into fallopian tube is called ovulation process. Now that egg will remain in the fallopian tube at this place for some days, checking for the sperm. If in that time the sperm enters, it fuses with the egg. Fertilization takes place in the fallopian tube. So fertilization takes place, zygote formation takes place. Then that zygote further in this tube itself, the zygote divides multiple times. That means a one cell to form 2 cells, 3 cells, 4 cells. No, so 1 will form 2, 2 will form 4, 4 will form 6, 8, 8 will form 16. Like that, the ball of cells is formed. That is what we call as embryo. Calyx, calyx, and finally forms it. The embryo is formed. So that embryo, now after formation of embryo, zygote converts into embryo. That embryo enters uterus. So in that duration of a time, before entering embryo into uterus, uterus makes this inner wall thicker. So uterus prepares itself by enlarging this side wall. Inner wall of the uterus becomes thick, and here it gains a more number of blood capillaries will be added here. Means they will be more, that inner wall, endometrium, will richly supplied with blood capillaries. So that is necessary for further growth of embryo. So every month, uterus prepares itself. After every 28 days, uterus prepares itself for that process. So the inner wall, endometrium, becomes thicker and it will be richly supplied with blood capillaries. If fertilization takes place, then that is useful. If fertilization won't take place, then again that wall shades off the blood which is present at this place, and the wall shades off which is not used. So it will move out. It has to be thrown out. So along with the that's egg which is not fertilized, this wall, inner wall, along with the much amount of blood is there, will be moved out of this vagina. That is called menstruation. And if fertilization takes place, then embryo formation happens. Then embryo will enter this uterus and stick to implant or stick to this wall of uterus. So that process is called implantation. And then after implantation, there is a growth of that embryo to fetus takes place in this uterus. Uterus enlarges, accommodate fetus, embryo will have now body parts, different organ development, tissue to organ system development takes place in this uterus. And for that thing, one more important tissue develops in this endometrium, that is called as, what is that tissue called? A special tissues required for the growth of fetus. That tissue is called as? No idea.
Mom, can you repeat the question?
Specialized tissue is formed in the endometrium for the growth of fetus is placenta. Placenta is very important part for life as well as for exam. Exam also they have asked every time 2 marks question on this. What is placenta? And how the this nutrition, what is the role of placenta in the growth during pregnancy. So placenta, structure of placenta also you need to know. Placenta is a disc-shaped tissue present in the wall of uterus. Is a disc-shaped tissue present in uterus. Now it has villi. It has many villi on fetus side and blood spaces on mother's side. Sir has shown you yesterday diagram. He has shown you the diagram of this one, villi and spaces, blood spaces. So placenta is a disc-shaped tissue present in the uterus. It has a richly supplied with blood. In this is the place where mother's blood and fetus blood comes in close contact. And through that blood, exchange of substances takes place. It has villi for the increased absorption. They have villi on the fetus side and the blood spaces on mother's side. So these 3 points you have to write for the structure of placenta. And then function of placenta or role of placenta. So how is the placenta helpful? What it does?
Ma'am, the impurities go from the baby to
What is impurity means here?
toxic waste.
There is
nitrogenous waste.
no word impurity.
Ma'am, nitrogenous waste.
Nitrogenous waste, you can write it as. So here, this is the place where the substances exchanged. So what are the substances exchanged here? The nutrients are transferred from mother's blood to fetus. Because fetus needs nutrients. So from where it will come? From mother's blood it will come. So nutrients will be transferred from mother to fetus. And then at the same time, nitrogenous waste is created. So those nitrogenous waste will be transferred from fetus to mother's blood. Second thing, oxygen. Now this one, the fetus is there. Fetus lungs are there, developed, but they won't work. Lungs won't work inside mother's uterus. So till that they are getting oxygen, the fetus getting oxygen through placenta. So oxygen from mother's blood will be transferred to fetus. And then the carbon dioxide will be produced. So that carbon dioxide will be again transferred from fetus to mother's blood. So this way, these 4 substances are exchanged through placenta.
Ma'am?
Yes?
Ma'am, why is the blood group of mothers and childs different?
That is depends on the structure, this DNA, genes.
Okay, ma'am.
Okay, but there is nothing happened that is a mother's blood is transferring to fetus. No, they have different in play at the placenta. The exchange of substances takes place between these two. They won't mix with each other. Mother's
So
blood and
only,
fetus blood won't mix with each other.
ma'am, so only nutrients is going.
Only nutrients are transferred. There is no exchange. What?
Ma'am, so blood groups can be different.
Yes, it is obviously. But blood group can be same, can be different. The blood transfer won't take place inside mother's this one uterus. There is no transfer of blood takes place between them. There is no mixing of blood takes place in both of them. Fetus and mother's blood, no mixing. They remain separate. Only they will comes in close contact near the placenta. So through the placenta, when there is a close contact, at that time the substances get exchanged by osmosis and diffusion. Clear?
Yes, ma'am.
So placenta.
Yes. Ma'am, successful reproduction is not possible between the cycles, right?
between these cycles. Yes, it is possible. After the ovulation, it is possible. It is possible. That time only it is possible. Okay. So important, very important question is drawing female reproductive system, different parts, functions to write the different function of the different parts, and then the structure, placenta function structure of placenta. Very important. Always asked 5 markers. Okay. This is very, very, very important part. Then the question is asked for 2 marks. What will happen? What are the changes will take place in uterus if the fertilization takes place and if the fertilization won't take place? So these are the questions you already have learned in class 8th as well. So what happens if the fertilization takes place? What are the changes takes place in the uterus when the fertilization takes place?
It becomes spongy and starts filling up with blood capillaries.
It will become thicker. Wall, inner lining of the uterus becomes thick and richly supplied with blood capillaries. And it will prepare itself to this implantation of embryo. And then if the fertilization won't take place, then
It will remain the same, nothing will happen.
No.
Lining will rupture and it will come down as
Shed off. We will use shed off.
off.
Now that egg dies because fertilization has not taken place, that egg is not of use. So that dead egg, the inner wall of the uterus is not useful, along with blood capillaries, all these things, mucus is also there. So that will shed off and bleeding starts. That is called as menstruation. And that will always takes place once in every 28 days. And at that time, the second ovary again now releases egg into fallopian tube. So this cycle continues. This cycle is called menstruation cycle that occurs in every 28 days. Right? Understood? And again, one more important question left out. Contraceptive methods. This one, the STDs. STDs, sexually transmitted diseases, the diseases which are transferred from infected person to healthy person through sexual contact. Those are of 2 types depending on there are the bacterial STDs and viral STDs. You have to remember name, always important, that always asked question. The bacterial STDs, name 2 bacterial STD. It is syphilis and yes, syphilis and gonorrhea. So it is gonorrhea and syphilis. And viral?
HIV,
AIDS and warts. Remember these 2. So 2 names, they always ask, right? Name 2 bacterial STDs and 2 viral STDs. Okay. So out of that, gonorrhea and syphilis can be cured by antibiotic medicines. Viral STDs could not be cured. And then to prevent this, not preventing only this, contraceptive methods. So what is the importance of contraceptive methods? How they are helpful? Yes. So contraceptive devices helps in preventing unwanted pregnancies, One. Second one,
Ma'am, healthy
population
children.
control. Yes.
Ma'am, healthy children.
Healthy children, no. So it is,
Ma'am, it prevents pregnancy. So how can you get healthy children?
that's what, that is not.
Population control.
Population control. First one is the prevention of unwanted pregnancies. Second one, population control. And the third one is this one, preventing STDs. Prevention of STDs. Now here question is asked, how is use of contraceptive devices, this one, it is now, what's it called? Helps in maintaining, what is this exact word, sentences are there? Give me a question, I have those questions. Health and prosperity of family. Health and prosperity of a family, yes. How are the contraceptive devices helps in maintaining prosperity and health of a family and health and prosperity of a society? Two questions.
Ma'am, could you repeat?
How are the contraceptive devices, contraceptive measures helps in our contraceptive devices helps in maintaining health and prosperity of a family? And the same question, in place of family, you can write society. These are two different questions. They have asked multiple times. Every year that question is asked.
Ma'am, could contraceptive devices stop bacterial STDs and viral?
Yes. Some of, not all. Some of them.
Ma'am, so one of the point could be prevention of viral and bacteria, and other can be,
this one, viral STDs.
yes ma'am, yes ma'am, and other could be less children, so less cost.
Less cost. Cost means here.
Ma'am, could you repeat the question?
How the use of contraceptive devices measures helps in improving health and prosperity of a family?
Ma'am, reduction in abortions.
That is, it is there how they are helpful for a family. So that is, they are helping in this one, avoiding unwanted pregnancies.
Ma'am, reduce economic burden, so better economic conditions.
Yes, so that you can write about the prevention of STDs, maintaining reproductive health of a women. And the third one thing, that is due to the lesser number of children, means less number of children, the smaller family size of the family less, smaller. So that is the parents are able to give all the resources, can made available all the resources to the children, and better lifestyle they can lead. Clear? Any of these points you can mention, right? Better lifestyle because of nuclear, less smaller size of a family. And about society, how it will be helpful in about society?
Less overall population.
Yes, it is there. We can write about the population control. And?
Ma'am, can it be less division of resources?
Okay, division of resources, right? You can do that one also. And the third one is preventing STDs. Right?
Ma'am, how will that benefit societies?
How it will not benefit the society?
Like only a family will be benefited. Okay, I got it.
Clear? So these questions they have asked multiple times. Sometimes they have asked family, sometimes they have asked society. So they keep on changing these words and they were asking. And the different methods of contraceptions. So you are not supposed to answer in examples. You have to write the methods. This is also always asked question, very commonly asked question in every exam. So you are not, remember again, students are only writing the examples. You are not supposed to write examples. You are need to write the methods. So first method is mechanical barrier method. Examples of this are condom and diaphragm. Second one is chemical method. Chemicals are intaken, that is oral pills. Oral pills will be taken too, and these oral pills helps in changing the hormonal balance. So they change the secretion of estrogen, and due to which the this one is ovulation is stopped. Third one is IUCD. IUCD, intrauterine contraceptive device. Examples are copper T and loop. And the fourth one, surgical. Surgical methods in that tubectomy and vasectomy. Tubectomy is done in female and vasectomy is done in males. In this one, the tube is cut and tied together again. So that is the transfer of male and female gametes will be prevented. So tubectomy, fallopian tube is cut and tied again. So ovary could not get released through, ovary could not release ovum. And even vasectomy also, vas deferens in males has been cut and tied together again. And then it is due to that there is a blockage. So sperms are blocked. So could not reach urethra. Clear? So these are surgical methods. So by surgery only that can be done.
Ma'am?
Yes?
Once there's a surgery done.
It can be regained. Some of that now it is the methods which can be, means it is, it can be this one. That tie has been removed and again this one will work out. It is not a permanent method. Earlier it was permanent, now it is not. It is a temporary solution. And one question is asked for 1 mark. Which of these methods is used to prevent STD? All methods are not used to prevent STDs. Only one method here it is used to
Barrier
prevent.
method.
Barrier method.
Only the barrier method. Only the barrier method is helpful in preventing STD.
And will the copper tea affect the health of women?
Yes, it will. Oral pill and copper tea both will help. This will affect the health of the person, health of the women.
Ma'am, which is more safe?
Barrier.
Okay, ma'am.
On the body. Okay, so these are all questions, important questions. Whatever the probable questions, all I have discussed, whichever asked multiple times in your exams. Okay, so all I have discussed from this chapter. This is a small chapter. If you think in the beginning, it is a big chapter. But when you learn 1 or 2 times, after that, it is not needed to read this chapter again. Because very common questions, multiple times, the same common questions are they have repeated. Okay. Any doubt anyone has?
Ma'am, I have a doubt in maths.
Okay, wait now. Are there any doubts you have?
No, ma'am.
No, ma'am.
Understood the chapter?
Yes, ma'am.
Yes, ma'am.
Now read chapter again. Okay, so that you will be, everything will be fixed in your brain and nothing, and now after that, no need to read again and again.